Hello and welcome to the GenomeSpace recipe webinar, How to Find Differentially Expressed Genes in RNA-Seq Data. Today we will be briefly introducing the topics that the recipe covers. Then we will demonstrate how to use the recipe in a live walkthrough. And finally we will take questions from the audience. This webinar will be recorded and posted to our YouTube channel shortly. If you would like to ask a question, please do so using the chat room, which you can access by clicking the chat icon. The recipe we will cover today answers questions such as, which genes are differentially expressed between my two phenotypes based on my RNA-Seq data? To use this recipe, we will need input data. We will be using raw RNA-Seq reads from multiple phenotypes, and we're going to be using a reference genome from the UCSC table browser. We will use GenePattern to align those RNA-Seq reads and determine which genes in the RNA-Seq data are differentially expressed and can tell the difference between our two phenotypes. Finally, we will visualize the aligned reads and the differential expression using IGV. So the basic idea here is how do we get from raw RNA-Seq data to a form of differential expression that we can actually visualize. So one thing to keep in mind as we go through this is that in this particular recipe, we're actually taking our raw RNA-Seq data and we are directly aligning it to our reference genome. In typical scenarios, you might actually want to go through a step where you check your RNA-Seq data to see if it is good quality before you do the alignment. And I'll show you some other recipes that are related to that, but in this particular recipe, we're going to assume that our data is high quality and we're going to jump right into the alignment portion. So these are the basic steps. We're going to have 3 data sets in total. 2 of them are available in GenomeSpace already, and I'll show you how to find them online. One will be a wild-type RNA-Seq data set, and another will be an RNA-Seq data set with a specific gene that has been knocked out. And we'll obtain a reference genome from the UCSC table browser. We'll send all of this data using GenomeSpace. We'll send it to GenePattern. And in GenePattern, we'll align the raw reads of each of the 2 phenotypes against the reference genome, and we'll determine which genes are differentially expressed within each phenotype, and then we'll compare the 2 to find which genes can distinguish between those 2 phenotypes. And at the same time, we'll also use other GenePattern modules to create an index file for our RNA-Seq reads. And we'll create a GCT file, and I'll explain what this is in a little bit, so that we can visualize all of our results finally in IGV. So now we'll do the live walkthrough. So this is GenomeSpace. This is a recipe resource. You can navigate to this page by going to recipes.genomspace.org. So in this case, there are a couple ways to search this. One, you can type in, for example, RNA-Seq data. The way that I'm going to show you right now is we're going to click List All Recipes. And because I'm interested in RNA-Seq data sets or recipes that cover RNA-Seq data, I'm going to filter by data type here, and I'm going to look for RNA-Seq. So you can see this is our collection of RNA-Seq recipes. The one we're going to be using today is Find Differentially Expressed Genes in RNA-Seq Data, but there are also other recipes you may want to use. So for example, as I mentioned, you might want to pre-process and quality check your RNA-Seq data before you do this recipe, if you're using your own data. If you, for example, ran an experiment and there were a couple of different times that you ran the experiment, or maybe used slightly different reagents, you might want to eliminate the batch effects in your RNA-Seq data. If you don't have your own RNA-Seq data and want to find RNA-Seq data online, you could find some using this recipe for identifying TCGA data. And then you can also do some extra visualization as well. But for now, we're going to just find those differentially expressed genes. So I'm going to click on this link. You can see this is the recipe. If you are more of a visual learner and you just want to watch the entire recipe, there's a series of 5 videos here that will walk you through the recipe. If you would like to know more about what this recipe is accomplishing, you can read the summary text. But for running the recipe, we're just going to skip right to the input section. So I can see here for the inputs that I need 2 data sets, this WT FASTQ-GZ and SFP1KO FASTQ-GZ. This is the wild type and the knocked out RNA-Seq data sets. And I can see by reading this Getting Data section that this is available in public recipe data, sequence data. So now if I go to GenomeSpace, so this is my GenomeSpace account. If I navigate to public, and scroll down, here we have recipe data, sequence data, and you can see here is that wild type and the SFP1KO FASTQ files. So at this point, what I could do is if I wanted to, I could copy this to my account. If I right click and click on copy move, I can choose my own account, and then I could click copy to copy that data. But this data might take up a little bit of room. So to save room, we're actually just going to be able to access this publicly right through the GenomeSpace interface through GenePattern, and so we won't actually have to copy this. So I'll show you how to do that. So if we take a look at this recipe, we can see that the basic steps are to start by getting our reference genome through UCSC table browser, then doing a lot of processing in GenePattern, and then visualizing in IGB. Let's get started with the first step. If I were more comfortable and more familiar with UCSC table browser, GenePattern, IGB, it is possible that I would just want to read this high-level explanation of what the step is. But if I were not as comfortable, I could expand these different steps, and I can see the detailed description of what that recipe step is. So in the interest of showing you how a recipe would work live if you were running this yourself, that's what I'm going to be doing, going through each step individually. So first, we need to use the UCSC table browser to retrieve reference gene annotations corresponding to the reference genome for our example data. If I go to the input section, I can learn a little bit about the data, and I see that it's using the yeast genome, Saccharomyces cerevisiae. So I also need a yeast reference genome. If you're using your own data, you might need a different type of genome, such as, for example, if you're using human data, you would want the human genome. So the first step, if I have these side by side, the first step is to launch UCSC table browser from GenomeSpace by clicking on the icon. So I can see here that this is a UCSC table browser icon. I can match it here. And if I click that icon, I'm now launching the UCSC table browser. I can also go through this text here to determine what parameters I need to change, or I can compare to the screenshots. So in this case, I'm going to look at this screenshot and compare it to what I see in UCSC. I see that I need the clade parameter to be set to other, and this will update automatically. You can see there's the genome is S. cerevisiae, the assembly is April 2011, the group is genes and gene predictions, the track is SGD genes, and the table is SGD gene, and we've selected genome. So all of those parameters match what's in this picture here. The output format, you can change to GTF, gene transfer format, and then you can select GenomeSpace. That's that checkbox. And in this case, I've already put in this name, which matches the name that's shown here, the SACSER3_SGD.GTF. So at this point, all I need to do is click the Get Output, and this will send the output to my GenomeSpace account. As you can see, it will refresh automatically once it's been completed. Then if I return to my GenomeSpace account and I reload my account by clicking on my username, I now have that GTF file. And this is that reference genome that we need to align our reads with. Or it's the gene annotation information. So now we've completed this particular step, and we can collapse this. And I see that the next step is to load data into GenePattern. So as I mentioned before, we're going to be loading the wild-type RNA-Seq data, the knockout version of the RNA-Seq data, and then this reference gene annotations. So there are 3 ways to do that, and I'm going to pick the third option, which is just to open the GenePattern tool from GenomeSpace and navigate to the GenomeSpace tab and then start using those files. So I'm just going to click on the GenePattern icon, and I can actually close UCSC now if I don't need it. I can click on the GenomeSpace tab here, and I should see my file under my username. So now I should be able to access all of that data. The next step here is to align the RNA-Seq reads, and what we're going to use for this is TopHat. TopHat is a very common tool used to align RNA-Seq data. It's a part of a suite of tools called the Tuxedo suite, and we're going to be using it to align the wild-type data and then also separately align the knocked-out data. So the first task is to change to the Modules tab and search for TopHat. You can see here that I've already run some jobs previously, and so those jobs are in my Recent Modules section. Just to follow along with the recipe, I'm going to search for TopHat. I can click on that module and see the TopHat tool. So just to let everyone know, as you're going through these different recipes online, it is possible that the screenshots may be out of date. So for example, this screenshot is showing TopHat version 8, whereas online right now I'm seeing TopHat version 8.1. If I would like to, I can continue using 8.1. That's perfectly acceptable. For this tutorial, I'm just going to switch it back to version 8. So as you're going through these recipes, please just check the screenshots. If they're out of date, please send us an email. You can email gs-help@broadinstitute.org. Or you can also leave a comment on that recipe page, and we'll try to update the screenshots as quickly as possible. So as I'm going through this, I'm comparing the screenshots to the tool that I'm seeing. So I can see here that I need to look for the bow tie index, and I need to look for reads pair 1. So the bow tie index parameter should be set to Saccharomyces cerevisiae, SACCER3 UCSC. So I can go ahead and do that. These are listed alphabetically, so it will be toward the bottom. And then reads pair 1 will end up being one of the 2 samples. So in this particular case, it's sfp1ko.fastq.gz. If I wanted to start with the wild type, I would use the WT fastq file. And the way that I can do this is I can navigate to the GenomeSpace account. And if you remember, I suggested not copying that data over, and instead we'll go through public. We'll find recipe data, and then we'll find sequence data, and we have that SFP1KO, and we can click it and drag it right into that parameter. If you forgot where this data was, you can always return to the input section at the top of the page, and it will describe where that data is located here. So next I need to have a GTF file. So if you look here on TopHat, you can see that there's a reads pair 1 and then there's a reads pair 2. RNA-Seq data comes generally in 2 forms. One is where the forward and reverse reads are in one file, and that's the type of data that we're using today, which is why we only need the reads pair 1. If you have the forward and reverse data sets or the forward and reverse reads in separate files, then you'll have an _1 or an _2 in your data, and you should load them into the separate reads pair 1, reads pair 2 sections. But today we'll just be using the reads pair 1 section. So if I scroll down, I can see I have this GTF file parameter that matches what I can see in this screenshot. So the first thing that I need to do is I need to click the Upload Your Own File button. I'm going to do that to create one of these click and drag boxes. And the GTF file is the same file that we got from UCSC Table Browser. So I'm going to scroll down until I can see my username, and then I can find that SACFAIR3 SGD file and click it and drag it into the GTF file section. Next, we're going to look for the Find Novel Junctions parameter. So if I keep scrolling down, it's located here, so we'll set this from the default yes and click no. And then finally, we want to change the output prefix. So this is the parameter that's all the way sort of down at the bottom, and it's already pre-populated. And what will happen is GenePattern will create an output prefix that will be added to the different outputs from this file, or from this tool. So what we're going to be actually doing is we're deleting that, and we're going to write in our own output prefix so it's a little bit easier to read. So I'm going to type in sfp1ko.reads, and that will be added to the beginning of every file. And then finally I can click Run to run TopHat. So this will submit the job to GenePattern. And if I look here, my job has been submitted to the GenePattern cluster, and it's currently in the pending stage. TopHat is a very computationally intensive tool, so even if it were running right now, it might take several minutes to run. And while that's running, we can do a couple of other things. So the first thing that we're going to do is we're going to run the second TopHat job. So right now we've aligned the reads from the knocked out samples to the reference genome, and now we need to align the wild type. So what you're going to do is click on the job and then click Reload Job. So instead of going through all the steps we already did, we just reloaded the job with all those existing parameters. And now all we need to do is just replace the file and replace the file name. So here, instead of SFP1KO, we want the wild type data. So the first thing that we're going to do is we're going to X this out to remove that data set, and then we'll click and drag the WT FASTQ file over to reads pair 1. And everything else will be the same except for that output prefix. So we want this to match the data set that we've entered. So instead of SFP1KO, we're going to change this to wild type. And now we can actually run this job as well. And now we can see that this is also pending, but the queue is much shorter. And if we switch to jobs, we can actually see that this TopHat job is actually running. You'll see here that I've already run a couple of other jobs as well. So one trick for working with GenePattern is if you want to see more jobs that you've run in the past, you can go to your user icon and then click My Settings. And then if you click on History, you can change this number to be a larger number. So I'm going to change it from 5 to 15. And then if I look at the job section again, I can see these previous TopHat jobs that I've already run. And the reason that I'm showing you that is because depending on how long this takes to run, we might actually have to use those TopHat jobs. So the next steps here, so now we've completed this TopHat part of the recipe, so we can collapse this. The next step is to identify differential expression in the RNA-Seq reads. So what this means is currently we have taken our wild type reads and our knocked out reads and we've aligned them to the reference genome. And now we need to use the cufflinks tool, specifically the cuffdiff module, to compare those two data sets and compare the wild type data set to the knocked out data set and see which genes distinguish between those two. And so we'll have to search for the cufflinks cuffdiff module. So we can start searching for that. So here we have cuff-diff. And if I compare this screenshot, I can see that the way that this works is I need to add both of these files into this module. And I'm going to be using the output from the previous top hat job. And I need to use both the output from the wild type run and also the knocked out run and compare the two of them. So first I'm going to describe a condition, and I'm going to start with the wild type. So we'll call this WT for wild type, and then we're going to add a file to this. And the file that we'll add is from the top hat job. So now I'm going to switch to the jobs tab, and unfortunately these jobs are not finished running at the moment, and again that's because top hat is extremely computationally intensive. So I'm going to scroll down to my previous top hat job, and I see here that I've run the wild type and the SFP1KO. And what we need to do is we need to use the wild type reads accepted hits.bam file. So I'm going to click that file and drag it over to this section. So now I've created my wild type file and condition. And the next thing I need to do is create the knocked out condition and file. So I need to click add another condition. And I'm going to type in SFP1KO for the name of my testing condition, and then I'll again scroll down and find that SFP1KO reads accepted hits.bam file, and click it and drag it into that section. So after this, I need to load a GTF file. If I scroll down, I can see that that's this parameter here. Again, I need to click upload your own file in order to create one of these click and drag boxes. And then I need to find this file in GenomeSpace. So again, this is the UCSC table browser file. So I can click it and drag it into that section. So next I need to change this library type parameter to FR unstranded. So if you're not sure what these different parameters do, you can always check the documentation. So at the very top of this cuff-diff module, you can see that there's a documentation link here. If you're not sure what these different parameters do, please read through that. That will help you identify what library you might have or what these different parameters might mean. So I need to change this to FR unstranded. And then I need to change the min alignment count. So this will be under the advanced input parameters. So you can see this is currently default set to 10. I'm going to change it to 0. This is just asking the minimum number of alignments in a specific locus that are needed to determine the difference between the two phenotypes. And then at this point, we can click Run right here. I'm going to scroll down and click Run. If I switch to my Jobs tab, I can see that this job has been submitted and is pending. So once this has run, next we need, what this will do is it'll generate a couple of different files. One of them will be this FPKM tracking to GCT file. So FPKM stands for fragments per kilobase of transcript per million mapped reads. And it's essentially just an estimate, so it's the RNA-Seq estimated expression for our RNA-Seq data. It's an estimate of gene expression. So the way that this will work is we need to convert that file to a more readable format, a GCT file, which will display the gene expression values in a matrix where each row is a gene and each column is a sample. So the first thing that we need to do is switch to the Modules tab and search for FPKM tracking to GCT. And we can load that. And if I scroll down, I can see that the input file here is this genes.fpkm tracking, which is the output from the CUFDIF module. So if I go back and I check to see under jobs if CUFDIF has finished running, it's still currently running, but luckily we have a previous version, and these are all identical. So I can look through this, I can see there's a couple different genes, and here we have genes.fpkm tracking. So there are many different files. So if I click and drag genes.fpkm tracking to the input file section, so this matches our parameters here in this screenshot, and at this point I can click Run to submit my job. So we'll click this Run button, and this will create a genes.gct file. So now we've completed the step in our analysis where we have compared the wild-type data set and the knocked-out data set, and we've determined which genes are different between those two data sets. And we've also created a new file type, or we will be creating a new file type that will let us visualize that much more easily. So at this point, you could potentially take that GCT file that will be generated. You could do other analyses with it. GCT files are often viewed as heat maps, and so you could actually visualize it as a heat map if you wanted to. In our particular example, we want to know not only the difference in the expression, so we want not only the GCT file, but we also want to see where the different reads aligned to match that expression. And the way that we're going to do that is we're going to visualize the aligned reads in IGV in addition to visualizing the gene expression data set in IGV. And so for that, we need to create a new data file called a BAM index file. And so we need to create indices for those aligned read files. So this is step number 5. So we're still working in GenePattern here, as you can see by the icon. So we're going to change to modules, and we're going to use the Picard suite of tools, specifically Picard Sort SAM. This will sort the files and create an index file. So if I compare this to my screenshot, I need to use one of those aligned reads files. So here we have the accepted hits.bam files, and we need to change the output format to BAM. So if I change back to jobs, so it looks like all my jobs have finished running. I can find, for example, the wild-type reads accepted hits.bam, click and drag it to input file, and then click BAM. We want the output format to match the input format in this case, and then we can click Run. So this will generate an index file for our wild-type sample, but we need to repeat this for the knocked-out sample. And so the quick and easy way to do that is to click that job and click Reload Job. And so here what we're going to do is remove this file and replace it with that SFP1KO accepted hits file. So if we scroll down and find that, we have sfp1ko.reads.accepted-hits BAM. And again, the output format is BAM to match the input format. And then we can click Run. So now we have our genes.gct, and we're running our two modules for creating our index files. So if we go back to the recipe, you can see that so far we've aligned all of our reads, we've identified the differential expression in those reads, and now we are in the process of creating our indices. So the next step is to save the files to GenomeSpace. And this lists 5 files that we need to save to GenomeSpace. So there are a few different ways to do this, but the way that I'm going to be doing it is to look in the Jobs view and just click on the files that I'm interested in. So genes.gct is one of those files. I'm going to click that file, and then I'm going to click Save to GenomeSpace. This will bring up a menu, and I should be able to select my username, and then if I click Select, it will send that file to my account. Let's give it a try with this spam file. Okay, that's uploading. So the BAM files are much larger than any of the other files, so they'll take a little bit longer to send. So you can see all of our jobs are currently finished. So first we have to send the genes.gct file, then the wild-type BAM file, the wild-type BAM index file, which has this BAI file name, and then the 2 knocked out samples. So we did wild-type BAM file, now we need to do the wild-type BAI file. You can save that. And that went much faster, so we'll do this SFT1KO file. And again, BAM files are quite large. They'll take a bit of time to send. And then the last file that we'll need to send, we have this BAI file that we need to send, and then genes.gct. So now these are all sent. So if I return to my GenomeSpace account, and again, if I click on my username to refresh everything, I should see genes.gct, and then this GTF files from our UCSC table browser that we've used throughout this recipe. And we have the BAM and BI files for both the knocked out samples and also the wild type. So now we have all those files that we need to visualize. So we can move on to step number 7. In step number 7, we've aligned everything, we've identified the genes that are differentially expressed, we've created our index files. Now we need to visualize everything. And to do this, we're going to be using the Integrative Genomics Viewer, IGV. And so what we're going to be doing is launching IGV from GenomeSpace. So IGV is a little bit different from the other tools that we've looked at in that it is a desktop tool and not a browser tool. So if I click on this IGV icon, now I'm going to be downloading an igv.jnlp file. So this will let me launch IGV from my computer. So here I have my desktop view. I have that igv.jnlp. If I double click that, this will bring up IGV. You might get a security warning, which you can ignore, so I'm going to accept the risk and run the application. You may also get some Java-related pop-ups. So for example, this is the Java console, which we can minimize and ignore. You might be asked to update your version of Java, but you'll eventually load IGV. So this is the IGV tool. You can see what's happened so far is that there's a menu up here. You can see there's a GenomeSpace icon or GenomeSpace menu item that I can access GenomeSpace from. There are a couple different sections, and I'll describe what these are. Right now, I have loaded and started IGV with that SACSER3 genome already loaded. If you're using human data, you might want to use HG19 or HG18, but I'm using yeast data, so I'm going to be using the Saccharomyces cerevisiae genome. If you have another data type, you can click on More and use a different genome. So if I compare this to the screenshot that I'm looking at, I see that in order to load all my files, I need to navigate to GenomeSpace, load file from GenomeSpace, and then at that point, I should be able to load the wild type accepted hits BAM file, the knocked out accepted hits BAM file, and the genes.gct. So I'm going to start with the genes.gct file. We'll go to GenomeSpace, load file from GenomeSpace. I'll navigate to my account and load genes.gct. You have to load each of these separately. So I can see here that this genes.gct file has the wild type expression and also the expression for the knocked out samples. Red indicates presence of expression, white indicates absence of expression, and we'll zoom in on that later. This is the gene annotation information for the reference genome. So now I'm going to load the wild type sample. So here I have both the BAM and the BAI file, but all I actually need to do is load the BAM file. IGV will automatically know, as long as you have these two files in the same folder, IGV will automatically know what index is being referenced. So I'm going to load the BAM file. And then I'm going to load the, so we just loaded the wild type one. Now I'm going to be loading the knocked out version. So I'm just going to, so I'm going to describe what these are. So this is the BAM files, and you can see that currently they're blank. And the reason for this is that IGV won't be able to render everything, so we have to actually zoom in order to see something. So if I continue scrolling down, I can see that at this point I need to zoom in on this specific gene that we know that we've knocked out. If I go all the way to the beginning and look at the inputs, I can learn that the sample that we're using is a Saccharomyces cerevisiae knockout of the SFP1 gene, which corresponds to the YLR403W gene in yeast. So I'm going to search for this YLRW gene, and what I'm going to do is up here in this box here, I can type YLR403W, and now IGV will zoom in on that location. So again, this is the gene expression. I can see here that in the wild type it's red, in the knockout version it's white. If I zoom in, and I can see that it says value, the wild type value is 9.46. The knockout value is 0, so this confirms that this gene was knocked out. Also, if you're looking at the reads themselves, you can see that these reads, there are a couple of aligned reads in the wild type sample, where there's no reads in the knocked out sample. So this confirms that we have knocked out that gene in our knockout sample. If I zoom out, you might be able to see other regions of the genome. So for example, this completely red region is a place where that gene is present in both the wild type and the knocked out version. This white area here is a place where that gene is actually absent in both the wild type and the knocked out version, or rather that locus. And then the gray areas are the areas where that gene expression was not determined. And so if I go to the very bottom of the recipe, or if I collapse this and then scroll down and compare my screenshot, I can see that I have this matching red and white pattern in the gene expression. If I zoom in a little bit more, I have a couple of reads that are aligned in wild type and no aligned reads in the knockout version, which is what we have here as well. And this results interpretation section will better explain what these different things mean if you want to go through that. So this concludes the live demonstration of the recipe. So now we're going to take some questions from the audience if anyone has any questions.